Respected judges and one and all gathered here, very good morning to one and all gathered here. I am Vishnu Shyam. I am going to present my research paper on genomic approach to identify association of environmental bisphenol A in daily use plastic as molecular disruptors in breast cancer. So, the last 6 decades have witnessed the introduction of various chemical compounds, bioactive synthetic compounds. One of the compounds introduced was bisphenol A. It is an endocrine environmental disruptor which is synthesized in 1905 and is used as a plasticization in various daily use plastics such as water bottles, thermal residues, children's toys and food containers. This BPA easily leaches out from the containers on varying temperature and pH, and studies reported it in human urine, blood samples and biological fluids. The World Health Organization and the FAO issued a safety tolerable daily intake of BPA as 1.5 to 4.2 microgram per kg body weight per day. So the active metabolite of BPA when it entered a bloodstream causes conformational changes in the homoiety of hemoglobin and aromatic amino acid residues such as trypsin, tryptophan, which is observed by spectroscopic methods. So this BPA acts via estrogen receptors and causes overexpression of MDM2 gene, which downregulates p53 tumor suppressor gene and induces cancer cell proliferation as reported in the human breast cancer cell lines. So the hypothesis of our project is shown in the flow diagram. The bisphenol causes overexpression of estrogen receptors, overexpression of MDM2 gene is reported in the breast cancer cell lines, and they suggest that there is a direct regulation of estrogen receptors by BPA and MDM2, which wholly leads to p53 downregulation, which causes increased risk of breast cancer. So our main objective is to investigate the association of bisphenol A, the active metabolite, and MDM2 SNP309 polymorphism with the risk of breast cancer. So after ethical clearance from the Institutional Ethical Committee, blood samples are collected from the breast cancer patients and control groups. The conformational changes were measured in the range of 200 to 800 nanometer by using UV visible spectrophotometry. The DNA genotyped by tetrams PCR. The primers are shown here, and the PCR products are analyzed by agarose electrophoresis. So this is the results in our study. The serum sample shows that the breast cancer patients had increased the mean absorbance at a range of 250 nanometer to 300 nanometer and 360 nanometer to 450 nanometer as compared to the control samples. So the table shows the statistical analysis for the association of MDM2 SNP309 genotype with breast cancer risk. So the allele frequency as shown here, the G allele, the variant allele is increasingly associated with the cases than the controls, which is statistically significant. This means there is the odds ratio is 2.17, that is 2.17 fold increased risk of breast cancer with the genotype G allele. So when coming to dominant phenotype, with reference to the TT as a control, the G in its dominant form increases the breast cancer risk by 2.8 fold, which is statistically significant. Whereas the TT genotype is present more in the control groups than the breast cancer patients, which is statistically significant with the odds ratio of 0.36. So we observed an increased mean absorbance in serum samples of breast cancer patients when compared to controls at this range, which is consistent with those results reported in previous studies. The peak absorbance at 410 nanometer due to hemoglobin is increased in breast cancer patients than controls, which indicate more free hemoglobin due to these conformational changes by BPAQ. Changes in amino acid residues in serum albumin and cytochrome C cause peak absorbance at 250 to 300 nanometer. So this MDM2 SNP309 G allele is increasingly associated with breast cancer patients, whereas T allele is increasingly associated with controls as shown in the table. The MDM2 G variant in its dominant form increases 2.8 fold increased risk of breast cancer, which is consistent with the previous studies reported. So there is an increased expression of estrogen receptors, presence of MDM2 G variant allele in human breast cancer cell lines in the presence of BPA reported by previous studies. So this suggests that the bisphenol A could be responsible for the mutation of MDM2 gene, a T2G variant, and causes overexpression of MDM2 protein, thereby leading to tumorigenesis. So the take-home message is, in conclusion, a significant association between MDM2 SNP309, a T2G variant allele, is associated with the risk of breast cancer in Indian women. The breast cancer patients have more BPK-induced changes than the control groups. Moreover, the direct correlation between the amount or level of BPA quantified and the breast cancer could not be ascertained with this small sample size. Further long-term studies with more sample size of controls and cases may throw some evidence on this. So the take-home message is, by reducing the daily exposure to environmental BPA, there would be a reduction in the increased risk of modern metabolic disorders and hormone-dependent cancers. So thank you for giving this opportunity.
Thank you, Sivam. Are you a doctoral student or a postdoc?
Undergraduate final year MBBS student.
Your final year?
MBBS student.
MBBS. Okay. Did you do any of these in vitro tests yourself, or you are presenting on behalf of a group?
Sir,
did you do these experiments yourself or presenting on
yes,
behalf
sir,
of?
it was my ICMR STS project, sir.
Oh, wonderful. So my question is very simple. You made a conclusion. Go back to your observations. First and foremost, who were your controls? Who were your controls?
Sir, controls are non-breast cancer, no history of breast cancer patient, no other any visible disease, sir, no cancer, sir.
Are they family members?
Sir? No, sir, no family members.
Why?
Sir, family members.
How do you know they are exposed to the same amount of bisphenol A?
Same locality, sir.
Within
the same locality, in Coimbatore.
In the same locality, two houses may have a different consumption. Yes, one may eat everything in a packet food format or in plastic bottles. The other may use only steel jug or a glass bottle. So it doesn't mean that they are exposed to the same level of bisphenol A. Can you go to one of your conclusions? How did you conclude that bisphenol A actually leads to a mutation? Go one step back. Yeah. How did you conclude that it causes a mutation in the MDM2 gene? Bisphenol, what a single nucleotide polymorphism actually shows is whether they were born inherently with that SNP. It is not induced by the environmental carcinogen. If it is a metabolizing enzyme, it just means that they are born with that SNP. Nowhere does it suggest that bisphenol A causes this SNP mutation. So in which case you have to prove that it has occurred in the breast tissue. It is absent constitutionally. Did you prove that?
We refer to the previous studies, sir. They proved that in human breast cancer cell lines like MCF7, in the presence of BPA, in the absence of BPA, they showed that BPA induces MDM2G variant and it increases the MDM2 protein, which leads to breast cancer.
So we need to know whether it is constitutional or in tumor tissue. You have done a constitutional study where you estimated the bisphenol A in the plasma. Am I right? Not in the tumor.
No, sir.
Okay. So where did you study the SNP? You studied it from the polymers in the blood.
Yes.
Am I right?
Yes, sir.
So you think it is a constitutional change, or they are born with that? It's a germline mutation, or is it a somatic mutation? How do you prove that?
So it could be a somatic mutation, sir.
Typically, SNPs are inherent. Yes. They are germline mutations. They are not induced by the environmental carcinogen. Okay, so it is very difficult to prove that the MDN gene mutation actually occurs. Unless you have, let us say, two people in the same family, all consuming the same food, whatever you are postulating, and in them you demonstrate that one of them actually shows the MDM2 gene mutation, the others don't show it. So that's the only way of using a hypothesis to prove that there is a. Typically, SNPs are germline. They don't mean anything. So, second postulation, what does this mutation actually do to bisphenol A? If you think that it is the endocrine disruptor, if bisphenol A was the endocrine disruptor and it caused an MDM2 gene mutation, what did it do actually?
So actually
the SNP. What did the SNP do?
The BPAQ active metabolite adducts to the DNA. It has two pathways, genomic pathway.
I know the pathway. So what is your postulation? Did the SNP can act as the same as the normal, non-mutated? It can increase or decrease. There are only three possibilities, no?
Yes, sir.
And typically, SNPs do not actually matter. You are you and I am me simply because of SNPs. Yes, that is the only difference, okay? Otherwise, everything else is common, right? So you need to prove that it actually increases, decreases, or remains the same as far as the metabolism of estrogen is concerned, or the expression of estrogen receptors is concerned. Okay? Right. Any question? Thank you.
Thank you.